Good day, everyone. Welcome again to Hematology lecture. So for today, we'll be discussing another topic, which is the hematopoiesis. So hematopoiesis, from the word or from the suffix poiesis, means its cell formation and development. And from the word or prefix hema or hemato, it means blood. So hematopoiesis literally means blood cell formation and development. So here are our objectives is to know and determine what is hematopoiesis and erythropoiesis. Next would be the thrombopoiesis and lastly leukopoiesis. So let's first discuss hematopoiesis in general. So we've discussed or we've mentioned it earlier that it is cellular formation, proliferation, differentiation, and maturation of blood cells. So we have different and various types of hematopoietic tissues. So these are organs and tissues or tissue areas in which the blood cells are produced or the regulation of production occurs. So we have the fetal hematopoiesis, we have also the spleen and the lymphatic tissue as the site for blood cell formation, and lastly, at birth and postnatally, we have our bone marrow. So we have several stages of hematopoiesis. We have three. We have first the mesoblastic stage, next is the hepatic stage, and the third stage should be the myeloid stage. So we'll be discussing them individually on the following slides. So let's proceed with the first stage, which is the mesoblastic stage. So take note of the colored text because they are very important for us to familiarize. So in the mesoblastic stage, the primary site of blood cell production is in the blood islands of the yolk sac of the human embryo on the 19th day of gestation. So again, the primary site in the mesoblastic stage is in the blood islands of the yolk sac of the human embryo. So it usually lasts or remain active for approximately 8 to 12 weeks. So the first blood cell that is formed in the mesoblastic stage is the erythroblast, which is the first blood cell formed by the first month of the embryonic life. So erythroblast is formed or developed in the blood islands of the yolk sac during the mesoblastic stage. So we have the three major hemoglobin that are produced during the mesoblastic stage. We have the Gower 1, the Portland, and the Gower 2. So during my college days, we have this mnemonic given by our professor. We have this Poseiga, Gozeiga, E. So to easily memorize the embryonic hemoglobin, so Portland has 2 zeta and 2 gamma. Next, we have the Gower. We have 2 zeta and 2 epsilon. And the Gower 2 is GAE, which is 2 alpha and 2 epsilon. So how do we differentiate the Gower 1 from the Gower 2? So I have this mnemonic GAE kote, GAE ku ti, GAE ko 2. So it's Gower 2, 2 alpha and 2 epsilon. So don't forget these mnemonics, the Poseiga, Gozei, GAE ko 2. Next, we have the hepatic stage, which is the second stage of hematopoiesis, or the blood cell formation. So from the word hepatic means the development or the formation of blood cells occurs in the liver. So in this case, it's in the fetal liver. So the blood cell formed here is erythrocytes. So in the mesoblastic stage, it's erythroblast. So we have additional cells. We have the granulocyte and the megakaryocyte at the third month of gestation, lymphocyte and monocyte at the fourth and fifth month of gestation, respectively. So we have additional cells. We have the GLM, so erythrocyte, granulocyte, and megakaryocyte, lymphocyte, and monocyte. So they say that in the mid-fetal life, the spleen and the lymph node has a role in hematopoiesis. So, but we all also know that in our spleen and lymph node, they have limited role as secondary lymphoid organs. So, based on the immunology and serology discussion, that we have two types of lymphoid organs. We have the primary lymphoid organs and the secondary lymphoid organs. So, Doc, what are the primary lymphoid organs. We have the thymus and the bone marrow. So what are the secondary lymphoid organs? So we have our spleen, our lymph node, our Peyer's patches, and etc. So they remain active in producing lymphocytes throughout the life. So, di ba, we all know that our T cells and B cells come from the bone marrow, and then the T cells mature in the lymph nodes. So that is why they are called secondary lymphoid organs. So in our mesoblastic stage, we have the three embryonic hemoglobin, the Portland, the Gower 1, and the Gower 2. In hepatic stage, we have our fetal hemoglobin, which usually occur at the fourth month of gestation, in the form of hemoglobin F. So I have this mnemonic to memorize the components of the hemoglobin F, which is FAG, hemoglobin F for fetal hemoglobin, 2 alpha, and 2 gamma. The third stage would be the myeloid stage. In week one, we've discussed about hematologic terms. We define myeloid as bone marrow or spinal cord. But in this case, myeloid means bone marrow. So hematopoiesis in this stage occurs primarily in the bone marrow. So hematopoiesis in the liver begins to decrease in this stage by the last trimester. So it is replaced by the bone marrow. So we have two additional hemoglobin in a form of adult hemoglobin. We have our hemoglobin A1, which has 2 alpha and 2 beta chains, and we have hemoglobin A2, 2 alpha and 2 delta. So majority of our adult hemoglobin is in the form of hemoglobin A1. Approximately 90 to 95% of our hemoglobin is in the form of hemoglobin A1. So 5 to 10% is hemoglobin A2, and we also have hemoglobin F in a smaller percent. So if our exam would ask what are the hemoglobin that can be seen in a typical adult, we have hemoglobin A1, which comprises the majority of the hemoglobin. We have hemoglobin A2 and hemoglobin F. So in postnatal hematopoiesis, typically at birth, the bone marrow is the only site of production and development of our blood cells, including the lymphocyte and other cells. So the hematopoiesis typically occur on flat bones. These are in the sternum, in our ribs, in our spine or vertebrae, in our skull or cranium, and our pelvis. In adults, they are typically seen or form in our sternum, in our chest here, in the sternum where our ribs attach or attaches, and we also have other flat bones. So just nice to know information that when we do bone marrow aspiration or bone marrow biopsy, we do it on the iliac crest. So specifically in the posterior superior iliac crest or iliac spine. So as you can see in the picture, the needle goes through the outer layer of the bone until it reaches the bone marrow space. So again, the location of our bone marrow aspiration is in the iliac crest, is in the posterior superior iliac crest. So another nice to know is when we do lumbar puncture to get CSF samples in cases of meningitis or we suspect infection in our uh, central nervous system, we do it on the level of the L3 to L4 or L4 to L5 because typically the spinal cord usually ends at L1 to L2. So it is best to puncture below that site for us to avoid complications such as puncturing the spinal nerves or the spinal cord. So next, so we have these bone marrow functions. So for the bone marrow to function, we have the three following cells. We have first the stem cells, second is the progenitor or the committed cells, we have the precursor cells. So we first discussed the stem cells. So take note of the red colored text because this words or statement can help us differentiate between the stem cell, progenitor cell, and the precursor cell. So for the stem cell, they are pluripotent or multipotent. Pluripotent or plural means it can differentiate into multiple or differentiate into any cell line. Again, it's pluripotent. It can differentiate into any cell line. So next would be a good example of which is the CFU spleen, or the colony forming unit spleen. So we have the progenitor cell, in which it is also called a committed cell. So it is unipotential, means it can differentiate only into one cell type. And then an example of which is the burst forming unit erythrocyte, the CFU erythrocyte, the colony forming unit megakaryocyte, and the granulocyte monocyte. So again, they are committed cell. They are unipotential and differentiate in only one cell line. Last would be the precursor cells. So the precursor cells are in a blast form. So they are usually the first identifiable stage of our series. We have the myeloblast for our WBC, we have the megakaryoblast for our platelets or thrombocytes, and erythroblast for our red blood cells. So again, we have the stem cells, which can differentiate into any cell line. Next, we have progenitor cells, which differentiate into only one cell line, and the blast form, which are the precursor cells. So here is a very good representation of the stem cells, progenitor cells, and the precursor cells. So we have the pluripotent stem cells in the form of the CFU spleen, and we have the following progenitors. And before going to the mature stages of cells, we have the blast. So now we'll be discussing erythropoiesis, or the red blood cell formation. So in this presentation, I will just briefly discuss or introduce to you the overview of each series. So for our erythropoiesis, or red blood cell formation, we have the following stages. We have the rubriblast, or the normoblast, and then go to the polychromatophilic normoblast, or the rubricite, and third would be the orthochromic normoblast, or the metarubricite. Meta means after the rubricite. And then the reticulocyte, or the diffusely basophilic erythrocyte, or also called the polychromatophilic erythrocyte. And lastly, the mature cell, which is the erythrocyte, or discocyte, also known as normocyte. So they say that the rubriblast produces up to 16 RBC. So usually the red blood cells has a lifespan of 120 days. As you can remember, in clinical chemistry, we have this test, hemoglobin A1C, or commonly called as glycated hemoglobin. So our glycated hemoglobin measures approximately, or the previous 2 to 3 months blood sugar, because it is dependent on the hemoglobin seen in the RBC. So the question now is, how much, or what is the lifespan, or the normal lifespan of an RBC? It is usually 120 days, or equivalent to 2 to 3 months. That is why our hemoglobin A1C measures the previous 2 to 3 months of our blood sugar. Next would be the polychromatophilic normoblast, or the rubricite. So other textbook would say that in this stage, this is the start of the hemoglobin synthesis, and also the last stage capable of mitosis, or division. Next will be the orthochromic normoblast, or the metarubricite. So in this stage, it is the youngest cell incapable of mitosis. And also the last stage, that is nucleated, because in our reticulocyte stage, usually we also, in here we have only fragments of the nucleus. And it is also the end of the hemoglobin synthesis. Since in our erythrocyte, or the mature red blood cells, there is no hemoglobin synthesis due to the absence of the mitochondria, and also the extrusion, or the disappearance of the nucleus, because a mature erythrocyte or discocyte has no nucleus. So if there is no nucleus and no mitochondria, which usually is responsible for the ATP production, so there would be no hemoglobin synthesis. So this is a very good picture of the erythroblast, or the erythrocyte series. So from the hematopoietic stem cells down to the burst forming to the colony forming and then to the pro-erythroblast to the RBC. So how to memorize the RBC series? We have this mnemonic PB4. We have the pro-erythroblast, the basophilic erythroblast, polychromatophilic or polychromatic erythroblast, orthochromatic erythroblast, retic, and the mature erythrocytes. Next, we go to thrombopoiesis or the platelet formation or production. So we know that platelets are not doesn't have nucleus because they are produced directly from the megakaryocyte cytoplasm. So usually the megakaryocyte make clusters of granules and then aggregate then to form the platelets. We have also this term the endomitosis in which there is nuclear division without cytoplasmic division. So they say that the megakaryoblast nucleus may contain 2 to 64 times chromosome, and each of the megakaryocytes produces 2,000 to 4,000 platelets. So take note, one megakaryocyte produces 2,000 to 4,000 platelets to make the normal range of our platelet in the peripheral blood, which is 150,000 to 450,000. Then we all know that we also have this hormone thrombopoietin, which stimulates megakaryopoiesis or thrombopoiesis or the megakaryocyte production. So TPO stimulates megakaryocyte production. So the maturation of the megakaryoblast usually takes 5 days. So next we have the mature platelets or the thrombocytes. Usually they measure 1 to 4 micrometer. And then they have a characteristic cytoplasm that is light blue to purple and is very granular. So our platelets also has granules. So the normal lifespan of a platelet in the circulation or in vivo is approximately 8 to 10 days. So why 8 to 10 days only? Because they are constantly removed by our macrophages in the spleen and the liver, so by the Kupffer cells. And when we have breaks in our skin, and then we have wound, so they are consumed by our coagulation cascade. So they are removed by the macrophage and removed or used by the coagulation. So take note of this. One third of our platelet is sequestered, meaning they are or they have a pool inside the spleen. So two-thirds or the majority of our platelet is seen in the peripheral blood. So in cases that our platelet or our patient rather has splenomegaly, meaning there is a giant spleen or big spleen, so the platelet count now would be low because if there is bigger spleen, so the spleen would sequester a lot of the platelets, thereby having the CBC platelet count at the lower level. So, in contrary, when the patient undergone splenectomy, meaning excision of the spleen, so there would be no spleen as a house or a pool for the platelet. So the platelet count now would be higher in the peripheral blood. So again, in patients with splenectomy, platelet count would be higher. In patients with splenomegaly, meaning giant spleen, the platelet count now would be lower. So next would be the leukopoiesis. We will first discuss the granulopoiesis, in which we have the granulocyte production. We have the neutrophil, the eosinophil, and the basophil. So usually the maturation sequence from the blast to the mature granulocyte in the peripheral blood would take approximately 14 days. So it is very important for us to know the characteristic of a mature granulocyte. Usually, the nuclear chromatin is more condensed. The nucleoli disappears, and from the abundant cytoplasmic granules, which are very basophilic, with non-specific granules, will become more scanty cytoplasm, meaning it will decrease its amount of the cytoplasm, with specific granules for the neutrophil, eosinophil, and basophil. So this discussion of the granules or the specific granules will be discussed in a future presentation. And also the nucleus are segmented and the overall size decreases. So next, here is just a brief or a summary of the granulocytic stages. We have from the myeloblast, which I mentioned before that it is the first recognizable stage in the bone marrow. Next, we have the promyelocyte, which is the first appearance of our primary granules. We have next the myelocyte, which is the appearance of the secondary granules, in contrary with the promyelocyte, which has the primary granules. And in myelocyte stage, it is the last stage capable of mitosis. And we have the metachromatic myelocyte, which is in the first stage, now incapable of mitosis. And we can see the youngest cell to appear normally in the peripheral blood, which is in the band or stub stage. And then, lastly, we have the mature neutrophil, eosinophil, and basophil. So from here, we have myeloblast, the promyelocyte, the myelocyte, the metachromatic myelocyte, and we have the band. So they are called band because they have no segmentation or thread-like structures that connects the lobes of the nucleus of the mature neutrophil, eosinophil, and basophil. So as you can see in the neutrophil, the mature neutrophil, they have segmented approximate, or they have segmented they are segmenters because they are connected by thread-like structures. The lobes are connected by threads. We have in neutrophils, they have 3 to 5 lobes connected by thin threads, and we have eosinophil, we have it is bilobe connected by a thin thread. Same with our basophils. So here are some pictures of the granulocytic series maturation. So next, we will be discussing briefly about T cell production. So this illustration came from my college lecture from Dr. Salaver. So he discussed very thoroughly the formation of the T cell. So let's first discuss a T cell formation. So T cell usually come from the bone marrow together with our B cells. So our T cell now will go to our thymus, where maturation of T cell occurs. So they will be now called the prothymocytes. We know that our thymus has two parts. We have the thymic cortex and the thymic medulla. So our prothymocyte will now go to our cortex, and they will now be called the double negative thymocytes. And then they eventually gain some markers or the CD markers, and they will now be called the double positive thymocytes. And then in the thymic cortex, they will now undergo this called the positive selection. So in the positive selection, we have in our thymic cortex thymic epithelial cells, where they will present antigens using class 1 and class 2 MHC. So for example, the thymic epithelial cells presented an antigen using a class 2 or class 2 MHC. So it should be recognized by CD4 T cells. So if the antigen is recognized by the CD4 positive T cells, it is um, accepted. But um, another example would be a class 1 MHC and it is recognized by the CD8, meaning it is functional. So they will now, or they won't undergo apoptosis. But in cases that our class 2 the cell presented using class 2 and the CD4 doesn't recognize that antigen, that CD4 positive T cells will undergo apoptosis or they will die. So the cells that pass through the positive selection will now be called single positive thymocytes or mature T cells. So the single positive thymocytes will now go or proceed to the medulla where there is negative selection. So again, in our thymic cortex, there is positive selection. In our medulla, there would be negative selection. There would be also their thymic epithelial cells. So in the medulla, the thymic epithelial cells uses this IREP protein or the autoimmune regulatory protein in which it enables the thymocytes or the thymus rather, thymus or thymic epithelial cells to express self-antigens. So for the negative selection, so for example, our thymic epithelial cells presented self-antigen using class 1. So if the CD8 doesn't recognize us or doesn't attack the self-antigen, so it passes the negative selection. For example, class 2, the thymic epithelial cells presented an self-antigen using class 2 MHC, and the CD4 doesn't react like in here. So it passes the negative selection. But in cases that our thymic epithelial cells presented using class 1 and the CD8 attacks or recognizes the self-antigen, so it will now undergo apoptosis. So we do not allow self-reactive T cells to mature and go out from the thymus because it could cause autoimmune disease or disorders. So from the 100% prothymocyte that entered the cortex down to the medulla, only 5 to 10% will go out or come out from the thymus to become or to be released in the peripheral blood. So that is how our T cells are produced in the body. So in summary, here are the nuclear characteristics of the cells during the maturation series. So we have the chromatin pattern from the smooth, homogeneous, delicate, or lacy pattern in here. They become smudgy clump until it becomes pycnotic or dense. Also, the nuclear shape changes from a bigger one, round or oval. They become more segmented in cases of WBC. They form lobes attached by or connected by fine threads. And also the nuclear size. From a large size, it becomes smaller. And there would be also extrusion of the nucleus. This is only true for the RBC. So in here at my back, we have a nucleus, and then at this stage, there would be an extrusion of the nucleus of the red blood cell to form the mature erythrocyte. And then again, the presence of the nucleoli. Immature cells has nucleoli, and then when they mature, they lose their nucleoli. So I think this is my last slide. So we have also an important note that in erythropoiesis, we are usually stimulated by erythropoietin. This hormone is produced in our kidneys. Also, erythropoiesis or the red blood cell formation is stimulated by hypoxia. So when there is decrease in oxygen level, so the bone marrow would interpret this as lack or decrease in red blood cells. So the bone marrow acts by releasing more red blood cells or reticulocyte in the peripheral blood. Next will be the leukopoiesis or the WBC formation. So they are usually stimulated by the CSF or the colony stimulating factors and also with the interleukins. And lastly, we have the megakaryopoiesis or the platelet formation, which is stimulated by a similar hormone called the TPO. This is similar to the EPO. The thrombopoietin stimulates platelet production together with our megakaryocyte colony stimulating factor. So there are many types of the colony stimulating factors. So I'm expecting that you will read that on our textbook for further readings. So I think this is my last slide. Do you have any questions? Thank you for listening.